All right, let's dive into one of the greatest scientific detective stories ever told. We're talking about the century-long hunt to find the actual blueprint of life itself.
You know, for basically all of human history, people knew that traits were passed down. You get your eye color from your parents, that kind of thing. But how? What was the actual molecule carrying that incredible instruction manual from one generation to the next? Well, that's the mystery we're going to solve today.
So every great investigation starts with just a few scattered clues, right? Long before anyone had even heard the term DNA, some seriously brilliant minds were already on the case, picking up the very first whispers of the trail.
Our story really kicks off back in the 1860s. You've got Gregor Mendel with his pea plants figuring out that some kind of invisible factors had to be passing traits along. Then, just a few years later, a guy named Friedrich Miescher is studying pus on old hospital bandages. Yeah, really. And he finds this weird stuff inside the white blood cells he calls a nuclein. Fast forward to the turn of the century, and Sutton and Boveri put two and two together, suggesting that these factors, what we'd now call genes, were sitting on chromosomes. The scene of the crime, it seemed, was inside the cell's nucleus.
Okay, now we get to a huge turning point. I mean, a real game changer. The year is 1928. And a scientist named Frederick Griffith stumbles onto a clue so bizarre it would completely flip the world of biology on its head.
So Griffith was working with two types of bacteria. One was totally harmless and the other was deadly. Here's the crazy part. He took the deadly bacteria, killed them, and mixed their guts with the live, harmless bacteria. And you won't believe it. The harmless bacteria suddenly became deadly. Something, he called it a transforming principle, was passing genetic information from the dead cells to the living ones. This was the smoking gun. It proved some substance could carry instructions. The only question was, what was it?
And that question just lit a fire under the entire scientific community. Suddenly, they had a full-blown whodunit on their hands, and two prime suspects emerged for the identity of this mysterious genetic courier.
In one corner, you have protein. Proteins are these super complex molecules made from 20 different amino acids. They do pretty much everything in a cell, so it just made sense that they'd be the ones carrying the complex code of life. And in the other corner, you have DNA. And DNA just seemed, well, kind of boring. It's only made of 4 repeating parts. Most scientists just figured it was structural scaffolding, like the studs in a wall, not the architect's blueprint. The smart money was definitely on protein.
But, as any good detective will tell you, you have to follow the evidence, not just what's popular. And a series of absolutely brilliant experiments started to pile up the forensic proof needed to nail the real culprit.
So in 1944, 3 scientists, Avery, McLeod, and McCarty, came up with this incredible experiment. It was basically a process of elimination. They used enzymes, which are like tiny molecular scissors, to destroy one suspect at a time. First, they used an enzyme to chop up all the proteins. Did the transformation still happen? Yep. Okay, so it's not protein. Next, they destroyed another molecule, RNA. Transformation still happened. But then they used an enzyme that only destroys DNA. And just like that, the transformation stopped, dead in its tracks.
Their conclusion, while written very carefully, was crystal clear. DNA had to be the transforming principle. Now, you'd think that would be it, right? Case closed. But the scientific world was still skeptical. It was a huge claim. They needed one last undeniable piece of evidence to really put this thing to bed.
And that final nail in the coffin came in 1952 from Alfred Hershey and Martha Chase. Their experiment was so elegant. They used viruses that infect bacteria, and they made 2 batches. In batch 1, they put a radioactive tag on the virus's protein coat. In batch 2, they tagged its DNA. Then they let the viruses do their thing and infect the bacteria. And when they checked to see where the radioactivity went, they found the tagged protein was left outside the cell, but the tagged DNA went right inside. That was it. Proof positive that DNA was the stuff carrying the instructions.
And with that, the debate was finally over. The case was officially closed. DNA, the simple, boring underdog molecule, was without a doubt the carrier of the genetic code.
But, of course, identifying the culprit is only half the battle. The next massive question was, how did it work? How could this simple-looking molecule hold the entire, unbelievably complex blueprint for a living thing? The final act of our detective story was just beginning.
Just 1 year later, 1953, James Watson and Francis Crick unveiled their now iconic double helix model. And this wasn't just a pretty picture, it was the ultimate revelation.
That elegant, twisted ladder structure immediately showed everyone how it worked. It could store massive amounts of information in the sequence of its rungs, and it could unzip down the middle to make perfect copies of itself. The culprit's methods were finally out in the open.
Discovering the structure was one thing, but actually reading the code? That was a whole other mountain to climb. The double helix gave us the key, and 50 years later, in 2003, we finally used it to unlock the whole thing. The Human Genome Project was completed, revealing the full sequence of our DNA, all 3 billion base pairs that make up the book of you and me.
I mean, think about it. This massive project was the true grand finale of this century-long detective story. We went from not even knowing what the blueprint was made of to finally being able to read every single word. Absolutely incredible.
So let's just quickly retrace the steps of this amazing journey. It all started with a simple question about Mendel's factors. Then came a mystifying clue, the transforming principle. The forensic evidence from Avery, Hershey, and Chase proved it was DNA. The code was cracked by Watson and Crick's double helix, and finally the entire blueprint was read. What a story.
And that brings us to today. Unlocking the secrets of DNA has opened the door to modern medicine, genetics, pretty much all of modern biology. The original investigation might be over, but the story is far from finished. Which leaves us with a really big question to think about. Now that we can read the book of life, what will we choose to write in the next chapter?